At the University of Washington in Seattle, scientists are researching how cell division impacts human disease. Their research can be used to understand the onset and progression of cancer at a mechanistic level.
If you want to understand how cells have become chromosomally unstable so that they make a mistake every 10 or 100 cell divisions, which is much more important for understanding human disease, then once again, you really need to be able to watch the cells over many cell divisions.
Tell us about research problems you experienced prior to using the new technology.
I and also my collaborator Holger Bastian approached James Ennis at the Institute for Stem Cell Research. We worked out all the techniques to culture the cells correctly and stain them using the robots down there. And we got beautiful images, and then the project basically screeched to a halt because we had no way to analyze our huge data set.
Analyzing large image data sets can be complex and labor-intensive, but GE Healthcare Life Sciences can help. Their InCarta image analysis software with Phenoglyphs machine learning classification simplifies cellular phenotyping from high-content screen data to greatly improve efficiency, accuracy, and ultimately speed to results.
You sat down in a microscope and start counting monasters versus, regular cells, interface cells. Usually it takes you like around 3 weeks to finish this counting. So basically with Phenoglyphs and InCarta, you can finish the experiment in 1 day compared with like 3 weeks that can take you with the regular system.
How did you like using the new Phenoglyphs module?
The most impressive thing I found watching the software in action was the ability of the user to, in a very intuitive way, train the machine. So you're presented with the images and you interpret them based on your training and then train the software. And when you're doing that, there's nothing to fear, in a sense. You're training the software to see what you see with your eye.
I think that the best thing about InCarta and Phenoglyphs is that it's easy to use. It's very intuitive, so with any other software, before using them, you need to read like full manuals of 100 pages, and that takes a lot of time. With Phenoglyphs, when you came here and installed the software, we were able to start analyzing data in like an hour. I would really trust to have like a technician, undergrad, graduate student doing those analyses, so it's really, really easy.
In addition to increasing lab productivity, what other challenges can this solution help address?
Both the granting agencies and the papers want us to address reproducibility and the sorts of things we do to increase the quality of our data set. And now we are able to do that. We can take a vast array of images, we can blind the data and then give it to somebody else and they can analyze it really fast. One of the things that we've just started using the InCell and the software to look at are to identify and find our CRISPR cells. So cells that we have modified so that certain proteins are expressed with a fluorescent label. And one of the things that's a little disturbing to a microscopist is that sometimes we can't see them. And so they're invisible, even though when they're at endogenous levels. So we have been using the InCell to find our little colonies of CRISPR cells, and it's fantastic.
Has this technology made your research easier?
Almost on a daily basis, we come up with more and more projects that we can use the high throughput image analysis. And so I think this kind of software and these kind of instruments will actually be adaptable in a variety of different labs. So they can add image analysis to their other types of analyses that they do. So I think it's going to be transformative in that respect.